Happy Friday, P-Bio. Today we're going to be wrapping up cellular respiration by talking about some variations in this metabolic process, some different ways that living things do cellular respiration, and a little bit about how this chemical pathway is regulated. So first of all, it's important to remember that every single living thing has to make ATP. All life has to do some form of cellular respiration so that they can make that energy molecule. So we're going to start by talking about how this happens in bacteria, in prokaryotic cells, because prokaryotes don't have mitochondria, right? They don't have the same organelles that eukaryotic cells do. So first, think a little bit about what we learned about the evolutionary history of mitochondria. Where did those organelles originally come from? If you remember, mitochondria originally were free-living prokaryotes. If you remember that endosymbiotic theory that talked about how a long time ago, early prokaryotes were kind of engulfed by other cells and eventually became these symbiotic organelles that we call mitochondria. So with that in mind, let's think about how these processes might be different in prokaryotes, or where they might occur in prokaryotes, because some bacteria actually carry out all these same steps: glycolysis, the citric acid cycle, the electron transport chain. So I just want to draw you a little picture here first. So if we started with this little bacteria, so here's our bacteria. The bacteria gets engulfed by another cell. Remember, this is what the endosymbiotic theory said. And so here it is engulfing that other cell. And that led to the formation of this organelle that we now have called the mitochondria. So here is our kind of modern day eukaryotic cell with this double membraned organelle called the mitochondria. So this is our mitochondria today. And if we think about how the mitochondria works and think about this process, right now, let's think about where these things happen in a eukaryotic cell. So this one is like our modern day eukaryotic cell. Glycolysis happens, so I'll label that step one, happens out here in, actually, I am going to enlarge this just a little bit. No, we'll be okay. Okay, glycolysis happens out here in the cytoplasm, right? The citric acid cycle, that second step, happens in the inside, in the matrix of the mitochondria. So it happens way in the middle there. The electron transport chain and the actual production of ATP, that ATP synthase enzyme, remember the electron transport chain is actually embedded in that inner membrane. So that's step three. Now, if we walk this back and think about where these membranes came from, remember this outer membrane here, this outer membrane around the outside of the mitochondria, came from this vesicle that engulfed the mitochondria to begin with, that engulfed this prokaryotic cell. The inner membrane is the actual membrane of the bacteria itself. So this membrane right here, this one, became this inner membrane of our mitochondria. So if we just kind of follow all of these back, if we peel off that outer membrane, way back here, so with a prokaryotic cell, with a bacteria, the differences aren't so much in the process, just in where they happen. Glycolysis, that first step, actually happens outside of, well, in bacteria, it would actually happen within the cytoplasm inside of the bacteria, because they take in the glucose, they break it down, and then step two is also going to happen inside of there, just like in a eukaryotic cell, it happened inside of the mitochondria in the very, very middle. But step three, remember that inner membrane was the bacteria's membrane. And so step three actually happens across the outer membrane of a prokaryotic cell, which means they use their electron transport chain, which is a bunch of proteins embedded in their cell membrane, to pump protons out of the cell. They don't have that intermembrane space. They just pump the protons right out into the surrounding area. So they increase the concentration of protons right outside of their cells. And those protons then diffuse back in through ATP synthase to make ATP in a prokaryotic cell. So the process in bacteria isn't actually different for those bacteria that carry out this entire aerobic process, but the locations are a little bit different. So I just wanted to kind of point that out so you can see they do still do the same process. The second variation we're going to talk about today is a really interesting kind of twist on this that pulls us back to the laws of thermodynamics a little bit. We're going to watch just the first couple minutes of this video called Plants That Keep Themselves Warm. And this is a way that some organisms, some types of plants, have taken this pathway of cellular respiration, but kind of altered it for a different purpose. So as you listen to this, think about, there's a part where he talks about skipping the ATP. Think about where in cellular respiration the ATP is made and what happens to the energy if it's not used to make ATP. You can also think a little bit about evolutionary benefits of this, and we'll talk about that in just a minute when this is done.
This is the checkers game where grandson and granddad will bond.
Being warm-blooded is really convenient. For one thing, you can go outside when it's snowing or hot without having to worry too much about your body processes shutting down. But not all organisms regulate their body temperature because it's a big drain on energy resources. It's mostly just birds, mammals, and some plants. A few hundred species of plants produce their own heat in a process called thermogenesis, and some of them use that heat to regulate their body temperature. It's based on the same idea as warm-bloodedness in animals. Energy can be released as heat. Plenty of your normal everyday metabolic processes like digestion release heat as a byproduct, but if that's not enough, you can also shiver, causing the chemical processes involved in muscle contractions to release more heat. When an animal can't shiver, like a newborn human baby or a hibernating bear, it can burn brown fat instead, a special kind of fat that's full of glucose and produces lots of heat when it's broken down. But plants can't shiver, and they don't have brown fat. So plants produce heat using another mechanism, a type of cellular respiration known as thermogenic respiration that takes place in their mitochondria. Mitochondria are usually described as the powerhouses of the cell, and for good reason. Their job is to store energy in the form of the molecule ATP. But in thermogenic respiration, the mitochondria skip the ATP and just release the energy as heat. At the most basic level, thermogenesis just keeps these plants warmer than their surroundings. But a handful of species, like the eastern skunk cabbage and the sacred lotus, take it one step further, with feedback mechanisms that they use to keep their body temperatures within a certain range, or thermoregulate. In thermoregulatory plants, decreasing internal temperatures triggers more cyanide-resistant respiration, keeping them warm. And if their temperatures get too high, they can just produce less heat and start to cool off. Now, thermogenesis might seem like a huge waste of energy, and it is, but for these plants, the advantages make the energy loss worth it. In some cases, it helps them avoid frost or keep snow away. The skunk cabbage, for example, can melt through snow, giving it early access to pollinating insects, while other plants are still buried. But many thermogenic plants are tropical, so they wouldn't have much reason to develop resistance to frost, and plenty of plants bloom just fine when it's cold out. Other plants are trying to attract pollinators, either by providing the perfect temperatures for mating insects or just by making themselves smellier. Like a dumpster on a hot day, a warmer plant will smell a lot worse. For instance, when a dead...
All right, we're going to stop there. He talks a little more there about one specific case, one specific example of how that's used to attract pollinators. So you noticed in there, he said these plants in the process of cellular respiration skip the ATP. And so if you remember, way at the end of the process of cellular respiration, the last part, that electron transport chain, pumps a bunch of protons out into the intermembrane space of the mitochondria. And as they diffuse back into the mitochondrial matrix through ATP synthase, that's what produces the ATP. The energy from that proton gradient gets transferred into molecules of ATP. But if you skip making ATP and just allow those hydrogen ions to diffuse back into the matrix, say through a proton channel in that membrane, that energy still has to go somewhere. Remember, the first law of thermodynamics says the amount of energy has to stay constant. It can't be created or destroyed. And so if that energy is not being used to make ATP, that energy gets released as heat. So it's a very slight variation on this process. They just take out that last step, but that allows these plants to produce more heat. And like they said, this does this takes a lot of energy. There's a big evolution or a big energy cost to these plants, but it also has big benefits that obviously have enough of an advantage that this trait was selected for in certain types of plants. So I just wanted to show you that kind of interesting variation on this process and a way that some organisms have adapted this metabolic pathway for a different use. They don't actually use this to produce usable energy in their cells. They use it to produce heat energy instead. Okay, so now we're going to go back to the idea of cellular respiration. But remember, up to this point, every type of cellular respiration we have been talking about has been aerobic cellular respiration that requires oxygen as that final electron acceptor in the electron transport chain. But there are quite a few organisms that can do cellular respiration even if oxygen is not present. And there are also a lot of organisms like us where our cells can continue to produce some ATP even if they're briefly deprived of oxygen. Like say when you're exercising super, super hard and your cells aren't getting enough oxygen quickly enough. So we're going to talk a little bit today about some variations that happen when oxygen is not there. Because if there's no oxygen, the electron transport chain would stop and back up, kind of just like what you saw in the case of the 7 deaths. When that final electron can't be given to oxygen, it backs up that whole chain and those molecules can't pass the electrons through, which means there's no proton gradient and ATP synthase can't make ATP. So it kind of stops the whole functioning of the mitochondria. There are a couple of ways that living things can produce ATP even without oxygen. One is what we call anaerobic respiration. The word anaerobic literally means without oxygen. And so this can happen. Some prokaryotes use a different final electron acceptor. They don't use oxygen, but they use another element that's also very electronegative to take those electrons at the end. So some prokaryotes that live, say, at the bottom of the ocean where there is no oxygen do this, and they use a sulfate ion instead of oxygen. Sulfur is also pretty electronegative, and so they use something different as that final electron acceptor. So the whole pathway is pretty much the same, but at the very end, that electron gets donated to a sulfate ion instead of oxygen. So that's one variation. The other way that living things can make energy without oxygen is through this process you've probably heard of called fermentation. Fermentation happens like this. The first step of cellular respiration, the glycolysis part that happens in the cytoplasm, that happens whether there's oxygen available or not, because it happens out in the cytoplasm, and that particular step by itself does not require oxygen. And if you remember, glycolysis does produce a little bit of ATP. It makes 2 molecules of ATP for every molecule of glucose that's split in half. But it also fills up some of these electron carriers, which are supposed to be taken over to the electron transport chain. But if there's no electron transport chain functioning, in order for glycolysis to continue happening, somehow this molecule has to be able to get rid of its hydrogen and go back to this oxidized form so that it can continue this process. And so the point of fermentation is to recycle that NADH back to NAD+ to allow glycolysis to continue so these cells can continue producing at least a little bit of ATP. And the way that happens is that those electrons on this hydrogen are transferred to the pyruvate or to molecules that come from pyruvate. So there's a couple different possible chemical pathways that can happen here. The first one is called alcohol fermentation. That's this top picture. So you can see here that glycolysis is still happening and it's still producing a little bit of energy, but it's filling up this NADH. And so in order for this to keep happening, we have to be able to recycle this. And so what happens in this case is that the pyruvate that's made when glucose is broken in half goes through this alternate chemical pathway over here, and on the way to becoming ethanol, a type of alcohol, it picks up those hydrogens, takes them off of that NADH so that it can be recycled and this process can continue. So the byproduct of that one is ethanol, this form of alcohol, and this type of fermentation is performed by bacteria and yeast. Our cells perform a type of fermentation called lactic acid fermentation. So it's the same idea. Glycolysis makes ATP, and it makes some of these NADH that then need to be emptied. In this case, the pyruvate molecule directly picks up that extra hydrogen and becomes lactic acid, becomes this molecule over here, which recycles those electron carriers so that glycolysis can continue. So these byproducts aren't necessarily desirable for cells. They're waste products that cells need to deal with, but this is kind of an emergency backup mechanism. Fermentation is not usually the primary way that these cells make energy, but it's kind of the emergency generator system. So even if there's not oxygen available, cells can switch over and do this for a while to continue making some energy until they again are able to get oxygen and return to normal aerobic respiration, which is way, way more efficient. So to put this all together, if we look at all these different pathways that we've talked about, the main one, aerobic respiration, goes through glycolysis, the citric acid cycle, and electron transport chain, which donates its electrons to oxygen. We talked about anaerobic respiration, where it's all the same, except some bacteria at the bottom of the ocean donate their electrons to sulfur at the end. And we talked about these two different types of fermentation. So it's probably pretty easy to notice here. The thing that is common to pretty much every single cell and every single living organism is the process of glycolysis. Anytime you see something that is common to many, many living things, the reason that it's found in so many different living things is most likely that it evolved a long time ago and has been preserved, has stayed part of those cells, even as other things, steps further down the pathway, have changed and evolved. That original step is still there. So the fact that this is common to all of these metabolic pathways tells us that this was probably used by really, really ancient cells, those really early prokaryotes that existed before oxygen was even present in the atmosphere. Those types of prokaryotes, and there are still some of these that exist on the planet, only do anaerobic respiration. They're called obligate anaerobes. They actually get poisoned by oxygen, so they have to stay far away from oxygen. Other types of bacteria have evolved that now only use oxygen. They're obligate aerobes. They need to have oxygen. And some of them, a facultative anaerobe, can kind of switch back and forth and do both. It will do aerobic respiration with oxygen and anaerobic respiration if there's no oxygen available. So the evidence that glycolysis is so evolutionarily ancient is, like we said, because it is so widespread, because every single cell does this, but also because it happens in the cytosol. This doesn't require any specialized parts or specialized organelles. So every single cell can do this process. It doesn't require any special equipment. The last topic we're going to talk about to kind of wrap up this part on cellular respiration is a little bit about the role of enzymes, both in catalyzing these reactions and in regulating how quickly cells produce energy. So this is some vocabulary we should already know about enzymes. Take a minute and review any of this that you need to. And then if you have your textbook handy, grab it and turn to page 146 and 147. Across the bottom of those two pages, you will see a very big diagram that shows the process of glycolysis. If you don't have your textbook, I'll put that figure up on the screen in just a second, but I want you to think about these three questions. Where does glycolysis happen and when during cellular respiration? explain the energy investment and energy payoff phrases, which you'll see on the top of that diagram, and you should be able to figure out what that means. And I also want you to look and see how many enzymes are shown in this process. So here is that full diagram. I had to kind of cut it in half, so this continues over to here. This shows the entire process, all of the chemical steps of glycolysis. And the first thing I want you to realize looking at this is how much you don't need to know about these processes. We just learned glycolysis is the first step. It happens in the cytosol. Glucose gets broken in half. It makes a little ATP and a little NADH. That's all we learned. But really, there are all of these different steps that happen just in that first part of glycolysis. Glycolysis is sometimes divided into two parts. You can see this top diagram is called the energy investment phase. And that should make sense if you look at this because 2 molecules of ATP actually have to be put in this first half to get this process started. But then the second half of glycolysis is called the energy payoff because you get ATP out. So these reactions here, this one releases 2 ATP and this one over here releases 2 more. So overall, you put 2 in and you get 4 out. So there's a net gain of 2 ATP molecules from the entire process of glycolysis. The other thing I asked you to look for and what we're really going to talk about here are the enzymes involved. Remember, the name of an enzyme usually ends in ASE. So that's how you can kind of identify these. But you also may remember that every chemical step, every reaction along the way is catalyzed by an enzyme. So just in the process of glycolysis, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different enzymes that each catalyze a unique step in this sequence of reactions. So enzymes are really, really important to making these chemical processes happen. And remember, this is just the first part. There are just as many enzymes that help with the citric acid cycle and further on through this process as well. But just in glycolysis, there are 10 different enzymes that help. We're going to learn a little more about this one right here, which also has a really fun name. This enzyme is called phosphofructokinase, which is just fun to say. And it's also really important as a way to regulate and control how quickly this whole process happens. So I am going to have you take a quick minute to read the information linked in the note. This is just a little 1-page thing about phosphofructokinase, about that particular enzyme. And I want you to pay attention to why this is important in regulating glycolysis and the idea of an allosteric regulator. So phosphofructokinase is an important regulator in this process because the step that's catalyzed by this enzyme is not reversible. Once that happens, the process continues, and it happens pretty early in the process of cellular respiration. So it's a good point to kind of regulate everything that comes after. Phosphofructokinase is what's called an allosteric regulator. And an allosteric regulator is a molecule that binds somewhere else on the enzyme. So if you remember, we often draw enzymes kind of like this, right? And this right here is the active site where the substrate molecule bonds. So this is the active site here. Where the substrate bonds, this is the substrate. And it catalyzes the reaction of that substrate, either building it up or breaking it down. But the way an allosteric regulator works is a lot of enzymes have another site somewhere over here where another molecule can bond. And so the allosteric regulator would be a molecule that comes and bonds over here. But the thing is, remember, anytime a molecule bonds to a protein, it's going to change the shape or the conformation of that whole protein. And so an allosteric regulator, when this bonds, it can change the shape of this protein such that the substrate doesn't fit anymore. Even by bonding over here, it changes the whole protein, so the active site changes shape as well. And now that substrate won't fit, and it can kind of deactivate that enzyme. in this case, in the case of phosphofructokinase, the inhibitor, the molecule that goes back and binds, is ATP. So once a cell has made lots and lots and lots of ATP, there's enough that it starts running into those enzymes. Remember, it's just floating and moving randomly around the cell, and it bonds with that enzyme way at the beginning step, which kind of turns off that enzyme and slows down the whole pathway, so less ATP is produced. But then as the cell uses up that ATP and the concentration of ATP in the cell decreases, then there's not enough and the cell needs to use that ATP instead of letting it bond to that enzyme. So it releases the enzyme becomes active again, and that pathway continues and it makes more ATP. So it's kind of a really clever way for cells to use the product of a reaction to control how that happens. So here's some definitions of that. An allosteric regulator is a molecule that binds to a regulatory site, not the active site, which changes the enzyme's shape, either making it more functional or, in this case, less functional. So here's an illustration of kind of how that works. This is the ATP binding site. And so when ATP bonds here, you can see it changes the shape of the substrate binding site so that it doesn't work as well. That's an inhibitory regulator. This one down here is a picture of a different regulator that would actually enhance this, that actually makes the active site fit better. But in this particular case, we're talking about this first one where ATP bonds and changes the shape such that it inhibits the activity of that enzyme. So this is an example of what's called feedback inhibition. The more ATP is produced, the more likely it is to bond to those enzymes and inhibit their activity, which slows down the process. And so it kind of regulates itself. This has been a lot of information over the last week or two. As you continue to work on your cellular respiration lab, your respirometer lab from yesterday, feel free to reach out to me with questions. Continue reviewing all of the notes from cellular respiration over the last week and a half or so. Continue working on mastering biology and reading your textbook, and let me know if there's anything you're confused about that you'd like me to clear up for you. I hope you have a wonderful weekend, and I will see you on Tuesday for an algae bead lab with the materials that you picked up at school this week. So I will see you then. Have a great weekend.